how do bacteria, other pathogenic organisms in food affect public health? What are the impacts on public health? And how can we reduce the occurrence of these illnesses? This study was conducted as part of the Feed the Future Innovation Lab for Livestock Systems. We also got funding from the Bill and Melinda Gates Foundation to particularly look at food safety and related infections. And both the Livestock Lab and the Bill and Melinda Gates Foundation have a focus on Ethiopia as a target country for their research. So that was the reason that we decided to work in Ethiopia as a country.
So I was very fortunate enough to travel to Ethiopia, I think it was a total of 8 times. We had a variety of samples that were collected. So from the 115 households that we enrolled, we ended up following longitudinally complete data from 106 households. So we were able to get monthly stool samples from infants as they were maturing up until 1 year of life, as well as from the household. So this included stool samples from the mother and the closest in age sibling, but also the surrounding livestock if that was present, and environmental samples such as water and soil.
I am from the place the study was conducted for, so I have a background in cultural background of the communities and local people there. So I was really the first line person to communicate to the community, to convince them to be involved in the studies, and I was conducting the enrollment and field work. So I have a good interaction with the communities, and especially in translation, the finding of the studies, also translation of the science to the community, so in a way the community understand it.
We used a variety of methods to analyze for Campylobacter. So we took a microbiology approach where we were culturing Campylobacter, as well as a series of molecular approaches. And this included polymerase chain reaction, known as PCR, as well as 2 sequencing approaches, whole genome sequencing and shotgun metagenomics as well. So when samples arrived to the lab, they were immediately processed. We logged in the samples just to track where was the source, where basically it was collected from, and what household. We then went ahead and extracted DNA from those samples. This was all done in Ethiopia, in Haramaya University. From there, we went ahead to test Campylobacter, and we did this in 2 different types of PCR approaches.
The impact that we are hoping to achieve in the long term would of course be that we are able to counsel the local population on how to prevent infections occurring in their infants. What we have now done, you could say, is diagnose the problem. So the next step would be to think about solutions for the problem. And so ultimately what we want to achieve is help people in those communities to keep their children healthier.
You gather these findings to then make some sort of intervention in the future that's going to help long term.